This is another episode of the Masters of Beautiful Achievements. What can we learn from them on how they apply natural science, systems thinking, and personal leadership to realize these beautiful achievements? My name is Alexander Princeton, and great you are listening to this episode. If you're looking for more inspiring episodes, check out my website, mastersofbeautifuliachievements.com. The link can be found here below. These conversations are made possible by our Patreon and Gumtree community, so I want to thank each of you for your monthly or even your one-time support. Your contributions enables me to create these amazing conversations, so thank you for your support. When you're not a member yet, click on the links below to join and be part of this amazing community. If you're interested in how I can help your business create beautiful achievements, choose one of my Gumtree offers on how you want to engage with me, or contact me directly via Scope Matters website. And now, it is time to enjoy the conversation with today's masters. So Fred, I'm really curious, what is biocomputing? Because I know computing, I know biology, but I have never heard that they have put these words together.
Well, biocomputing is actually computing with biological elements.
Mm-hmm.
And there are not that many ways to do this, okay? Some people are maybe using bacteria, for instance.
Mm-hmm.
On our side, we are using human neurons for computation.
But then how do you use living cells for a computer? Because I know most computers are using silicons with electrons, and we put a current in, and then we have a processor, and then it does what we want. And how would that function with biology?
Maybe you have to take a step back here because you're right on what you said, okay? But if you consider ChatGPT, for instance,
Yes.
an image recognition system based on AI, for instance,
Mm-hmm.
would you say this system is computing?
I have no idea what the definition of computing is. Processing, I don't know if that is the same as computing.
Well, actually, it processes information. And
Yeah.
in order to process information, it actually computes simulations of neurons.
Okay.
This is how the system works. So we can process information with artificial neurons, and we can process information with living neurons. This is exactly what you are doing right now with me.
Yeah, for sure. Today I have Fred Jordan in the show. He is, do you say founder or director from Final Spark?
Yeah, co-founder.
Co-founder. Could you explain a little bit more about you yourself and how you ended up with Final Spark?
Well, I'm a French physicist and went to Switzerland to make a PhD in signal processing. Founded the first company, which I'm still running, and founded another one where the goal was to create something different in AI. Really start from a white piece of paper and make totally different assumptions.
Mm-hmm. And that has become Final Spark with biocomputing.
Well, at the beginning, it was not biocomputing. We were looking at different ways to sort of make a revolution in AI, okay?
Mm-hmm.
And one of the directions that we were looking at a lot, among many things, was to simulate artificial neurons in a way that was way more realistic.
Mm-hmm.
That means temporal simulation, things like this. And doing this, we realized that the power consumption of the simulation was really huge. At this point, we realized that maybe if we use living neurons, then it may be more power efficient.
Mm-hmm.
And as a matter of fact, the difference of energy efficiency is about 1 million in favor of living neurons instead of artificial neurons.
That is quite a lot, if I assume.
Yes, it is. Yeah. So, I mean, from an engineering perspective, it was simply the best solution to use living neurons. Particularly when you think about AI today. AI is run using artificial neurons. And there is an energy problem that is coming with the vast deployment of AI. So if you can replace artificial neurons with living neurons and be 1 million times more efficient, you are going to solve definitely a problem.
And that was the reason why you wanted to move in this direction, or was it also links to, for instance, neuroscience? Because working with living neurons is quite exciting with what I've read today.
I must admit that initially, no, I was not particularly interested by biology. I would
Mm-hmm.
say even not at all. And the reason is that I was an expert, in AI programming, maybe physics, and it took, I don't know, 30 years to get this level.
Mm-hmm.
And when I realized that we had to move to biology, I was not that happy actually, because I was a plain rookie again. I had to learn
Yeah.
absolutely. So it was absolutely not a choice by preference. It was a choice by engineering. And just for power efficiency perspective, if I wanted to have realistic neurons performing computation, I needed to go to biology.
Yeah,
So
because
this was just the best solution around.
because one of the things I read, I think it was a couple of years ago, that DNA can store, I don't know, 1 trillion or a huge amount of information, or even in a cell. I don't know what that information was, but biology can store much more terabyte data in a very small space than what we have now in artificial intelligence of storage. I mean, is that also something that comes into the equation? The biology can store much more information than human created technology.
No, that was not the main driver. The driver was about processing information
Mm-hmm.
in an efficient manner. I mean, efficient, I mean, by energy perspective. And the fact is that neurons are the result of 300 million years of evolution.
And for the speaker, where do we find neurons? I mean, in humans, in animals, in plants, everywhere?
So you mean the neurons that we use at
No,
Viral Spark?
no. In general, what I mean, where do
So
we find neurons in life?
So it's really in nervous systems. Okay, so you will find this in animals, in insects. You will
Mm-hmm.
find neurons. The nervous system can be a central nervous system, as for you and I, and I,
Mm-hmm.
or it can be distributed across the animal, like for some insects, for instance.
And what does a neuron do then? So why is it so good as a computation machine?
So you are asking an ambiguous question here, okay? Because you ask me, why is it so good as a computation machine? But I have two ways to interpret your question. You can ask me why it is so efficient as computing, or why is information processing using neurons good? And
Let's do them both.
okay, so probably I'm going to disappoint on this because for the first thing, I mean, the power efficiency, I don't understand. I don't know why it's that power efficient. Okay. A number of people have mentioned that the fact that real neurons are spiking, so they
Mm-hmm.
are not active most of the time, may be one element. But this assumption doesn't may explain only a fraction of the power efficiency. Okay, because we have neuromorphic chips, for instance, which are done with discrete electronic components to simulate neurons, which are also spiking, but they are by far not as efficient as living neurons. Okay, so as I said, it's the result of 300 million years of evolution where efficiency was the main goal. So, of course, at the end, nature created something efficient. Now, for the second question, why is processing of information well done using a collection of neurons? So, this is a very profound question. Okay, very profound. It's and I don't have the answer. Okay, but just some guidelines. There is something called connectionism,
Mm-hmm.
and you can find on the internet about connectionism. This is really a science topic in processing of information. When you consider that if you create entities which are connected together, they are able to process information in an outstanding way. And this is exactly what's happening with AI today.
Mm-hmm.
AI is a collection of artificial neurons which are collectively processing information. Why is this way of processing information so um, um, so good? Okay, and I don't know. I don't know the fundamental answer to this question.
And how are you then able to, because that makes me curious in two ways. One, you put a neuron, you have put a neuron on a chip, and it communicates with a computer, which is quite mind blowing. And secondly, how do neurons actually work in real life in our brain? So let's.
So, yeah, for the first question, normally, most of the time, we don't put one single neuron, okay? We create what is called brain organoids, which is
Mm-hmm.
going to be a small organ. So, organoid means like it's a bit like an organ, okay?
Okay. Yeah.
And that will be, this organ will be a specific area of your brain. So we select which area of the brain we want to reproduce, and we are going to reproduce a small part, for instance, of your cortex or your striatum or hippocampus. Okay, we say, okay, we want to reproduce this, and we are going to reproduce, and it's going to be a small ball, like a half a millimeter of diameter, and
Yeah.
this is what we put on the electrodes.
Wow. Is there a difference between the different neurons in our brain that actually can use on your in your system?
You mean, of course, well, the neurons in the cortical neurons, for instance,
Yeah.
are indeed different and they connect in a way which are different between them. So this is why it's important.
So in our brain we have different types of neurons that all have a different function to let the whole let our brain operate. And you are able to pick out those neurons. I don't know. Is there a specific neuron that actually is working in your technology?
Yeah, we have very specific neurons. You know, there are about 400 types and subtypes of neurons in the brain. So we are selecting very specific area of the brain and to create these organoids. And that's not enough. We even connect. So we can do, for instance, cortical neurons, um, and then connect or dorsal cortical neurons, which are this part of the brain, and then connect to hippocampal neurons and create a so-called assembloids, which are two brain organoids together, connected
Mm-hmm.
together. So,
And what happens then?
so, so what happens first is that we have hopes. Each
Yeah.
time we do this, okay. This is a new field, okay. You can even find some publication even this year about 2025 about assembloids and how to connect different types of organoids. And um, there is what we hope and what is documented in the literature so far, the very small literature that exists.
Mm-hmm.
So what is documented is that if you are different part of the brain and you connect those brain organoids, you're going to have possibly some oscillations of activity between the different areas of organoids that you've connected. In our case, we have specific reason to connect them because we want sensitivity to some neuromodulators, for instance,
Mm-hmm.
um, or sensitivity or ability to spike. So this is why we are going to construct this as assembloids. So assembloids, it's really what it means. It's you are assembling some organoids. So the word assembloids, which we have not created. So
And that means that you stimulate how the, let's say, let's see if I have the right words, the dentitus and the axons, how they connect together, which is basically the receptors and the transmitters.
So there are different things in what you're saying. Okay, so indeed we are going to stimulate the neurons. Okay, and so there are two ways to stimulate the neurons. Okay, I'm going to start with the simple one, which is electrical stimulation, in which we are going to send a very small current, and if you do this, the neurons is going sometimes to answer, to respond to you with a spike of activity, very
Yeah.
small spike of 3
Yes.
milliseconds. And so this is the primary way to stimulate the neurons is by electricity. Now, indeed, they are connected together. And the way they connect is that the neurons are going to send this spike of activity to other neurons using their axon, you're right. And the other neurons are going to receive the information from the axon using their dendrites. And the connection between the dendrites and the axons is done using a very, very small interface, very small, very, very small ball, which is called the synapse. Okay. And everything happens in this synapse. The learning and everything is there in the synapse or in additional or removing synapses.
That's amazing, and that happened. That I'm just, while you're talking, I'm thinking about my own brain and seeing the videos online how information is transferred in our brains, or how our brain transfers signals to the rest of our body. And there is also talks that the neurons can reconnect themselves to other ones if the situation arises, that that they disconnect the synapse and connect it to another neuron. I mean, is that something? I mean,
It's happening. No, it's happening all the time. Okay, it's happening.
they make their own pathways depending on efficiency or needs.
It's absolutely required that they reconnect, okay, or they disconnect. Okay, if you want to learn something, you have to do this. There is no other way, as we know.
What is your background, Fred?
So I'm more a physicist,
Yeah,
and my PhD is in signal processing. So I would say applied mathematics.
and now you're moving in neuroscience. From a physical point of view, what has been the learnings for you in the last years?
Well, it's been huge because I really started from scratch. Okay,
Mm-hmm.
you cannot imagine my level of biology was really zero. Okay, okay, and we when we decided okay, we are going to work in biocomputing, we had to learn how to do culture of cells. Okay, cell
Mm-hmm.
culture. Okay, so how do you take some neurons from liquid nitrogen, put them to room temperature, and multiply them and make them happy, and create brain organoids with this. Okay, so there was a lot of things to learn, but for some reasons we met a lot of people who were happy, basically for free, to teach us. So we met some professors and a number of people who spent a lot of times, particularly in cell culture,
Mm-hmm.
because in cell culture you're doing this with your hands. Okay, you've got a so-called laminar flow hood where you put your hands below and then you start manipulations. You have to use pipettes and things like this. So we didn't know anything. So it's not only the theory that we didn't know. We didn't know neither the practice. So
Mm-hmm.
and we had to learn a bit of both. And we spent 1 year about this, and then we bought all the hardware to create our own lab, because during the first year we had the ability to use an existing lab and see and learn in practice what has to be used and how you do this.
And what else have you learned in the meantime? I mean, what kind of insights on neuron
Well,
and
there are a number of incredible things that I discovered. I discovered it's everything for me was a discovery, because I never, I didn't know anything. Okay? It was not a discovery for the experts, but when you start cell culture with neurons, okay? First thing is that you learn that neurons are going to stick on plastic surface, okay? And this is the way you multiply them, okay? So at the beginning, you start with, well, for instance, 100,000 neurons that you have taken to room temperature from liquid nitrogen, and that are very specific protocol. And you are wrong here; everything is dead, okay? And you are going to put this in plastic, on the plastic, and you always use what is called a medium, which is basically water plus glucoses, amino acids, salts, etc. So it's a mixture that you buy, okay? And they are going to adhere, to stick to the plastic, and to multiply. And it takes some time. But if you take 24 hours, you come back, you go to the microscope, you will see there are more and more neurons. And neurons want to connect spontaneously to each other. You were talking about dendrites and axons before. Okay. You can find on the internet an incredible video that shows that when they are on the plastic in cell culture wetware, what they do is that if they want to get closer to each other, they are going to use their dendrites to walk on the surface of the plastic.
No way.
Like legs, okay? And of course, dendrites are not meant to be like legs, but they are used by this. So this is an interesting thing. If a neuron is isolated from the rest, it's going to die most of the time. And remember that we are using stem cells, okay, neural stem cells. So these are not actually neurons. These are wannabe, the wannabe neurons. So as long as they are stem cells, they can multiply, I would say, infinitely. And this is for free, okay? If you create integrated circuits with transistors, transistors are not going to double for free overnight, okay? Here's the case, okay? It's going to multiply. And at some point, if there are too many of these neurons on your plastic system, then they are going suddenly to stop multiplying, and they are going to differentiate. So differentiation is when you lose your ability to multiply yourself as a stem cells, and you actually become a real neuron. This is going to take some time, a few months actually, for a good differentiation. But after a few months, spontaneously, a mature neuron is going to spike. That means it's going to create some so-called action potential, which are a small discharge of electricity. Actually, it's not really electricity, but we can measure it using electricity. And this is also a spontaneous behavior, and they will connect together spontaneously and start to exchange spikes of electricity. So this is also quite magical, I would say.
Yeah, I mean, I just have a son of 4 years old, and I mean, the way you're talking about stem cells is actually how humans get born, and I think that is very similar to how a brain grows. I mean, does a stem cell know what it wants to become?
That's a very good question. A very good question, actually.
That is the beginning of everything.
Yeah. So there are different kinds of stem cells. Okay. There are the king of stem cells. Okay, which are called the pluripotent stem cells. Okay, and I'm happy to talk about it because we just started to work with those. So the king of stem cells, pluripotent, means they are potentially everything. Pluri. Okay, they can become everything they want. Okay, so if you have pluripotent stem cells, you can do neurons, but you can do also skin or eye, hair, whatever you want. Okay. And yes, you're right. There are specific protocols. So protocols means partly molecules, okay, proteins that you have to put in your medium, so in the water where there are the cells, to force them in the direction that you want them to become. And this is what we do. Anyway, so we know exactly. So in biology, if you look, you can look on the internet. All the protocols are published and available for free. If you want to create cells of any part of your body, you will find a protocol for this, for the main parts, okay? And even for the brain, the protocols are different for the different areas of the brain. This is why we are going to choose different molecules and sequencing of molecules to guide the neurons in this specific type of first neural stem cells. So neural stem cells is in between, is a stem cell which is not pluripotent anymore because it's neural. So its destiny will be to become a neuron. But which kind of neurons from which part of the brain, then you decide with another protocol. So 2 protocols, one for neural stem cells and one for which part of the brain.
And that means that the proteins are amino acids, and based on the amino acids they get as a feedstock, they decide what kind of cell they become.
Absolutely, yeah. And you can buy those, yes.
Interesting. I'm just processing it because that's sort of like, is this a sort of like, we move into science fiction? Is this sort of the beginning of a humanoid robot that has a working biological based brain? Because that is sort of based on your information, my mind moves towards.
Well, there is something around these lines actually. That what we are trying to do is something around this. We are trying to resynthesize a nervous system that can learn and process information. And now, how do you use it? Okay, you can put it on a robot if you want, yeah, but you can put it in a server and replace, for instance, Amazon Web Services, which is serving ChatGPT, with this one who will be doing also some AI, for instance. For the rest of the world, it will look like AI, but inside it will be, some people say OI, organoid intelligence.
Yeah, and how far is consciousness then away from how neurons operate? Because what I understand with what I've read is if you have a lot of neurons together, like our brain, it stores information in themselves somewhere, I assume, and I mean, it then becomes sort of a. I mean, let's rephrase it. If it gets a life of its own, it's sort of like our brain. Every person is wired differently, and has different capacities. What happens if you put a lot of neurons of different, let's say, species together? Will it then move into consciousness? Because I mean, then we also talk about what is information and how it's processing information out, and where does consciousness come from?
So, well, I don't have answer for this, okay? Because first, I don't know what consciousness is. It's not actually well defined.
Mm-hmm.
And, it may not even be something like 0 or 1. There may be some degree of consciousness. And is there a critical mass of neurons where you get consciousness or not? I don't know. And this is a topic that we may be able to help indirectly by bringing science, but I don't have any expertise on this.
No, I understand, but it's just as I mentioned, it's when we compile everything, you see certain direction where things can move, and I'm just let's rephrase it. I'm just curious, how do you then use the neurons as a computing power? Because you need those. Is it that you turn it eventually? You turn it into 0 and 1, I assume, for a computer output. And how do you use biology to put it into a 0 and a 1?
But the same way as you use an artificial neural network, you know that the output of an artificial neural network is not 0 and 1.
I have no idea about artificial
I tell you.
intelligence, Fred.
I tell you,
Well,
people, tend to think that artificial neural networks and AI is binary. It's not at all, okay?
Okay.
So there is no fundamental difference in the in the type of output of an artificial neural network and biological neural networks. I would say fundamental, okay? The main striking difference is that biological neural networks are spiking. That means as time goes by, there is nothing, and then there is a spike, and nothing again. Okay. Normally, most of the time in AI, we don't use this kind of simulations. It's too complicated.
Mm-hmm. And you can have a higher density of neurons based opposite to the current artificial intelligence. I mean, let's say per square centimeter. Just
Can we?
curious.
I don't know. I don't know if we can have a higher density of living neurons compared to artificial neurons. I'm not able to answer to this question. However, what I can say is that in the case of brain organoids, so this is what we are doing. We are connecting in 3 dimension. Okay, our brain is in 3 dimension also. Okay, and I mean, physically speaking, when most of the integrated circuit and processors are done in 2D.
Yeah.
Okay, so we have sort of an advantage of going 3D because we have all the connections which are in 3 dimensions and are done spontaneously. So this is one of the striking difference from an architecture point of view between artificial neurons and biological neurons.
For my clarity, what is an artificial neuron? Because when you talk about artificial intelligence, I think about hard drives and software. So maybe you need to lighten me up.
So artificial neurons, so first, it's what is used in AI basically everywhere today. Okay? It's what is used on your iPhone to sort pictures of cars or whatever, what is used in ChatGPT and in every art generation, automatic AI art generation program and picture generation program, etc. Okay. So, and what is it? It's very simple. It's just mathematical formula. Okay.
Okay.
That works this way. If you have one neuron connected to 10 other neurons coming to it, okay, it says that this neuron will have a state which is defined by the state of these 10 neurons multiplied by their 10 connections. And the state of these neurons, if all what comes is positive, then the state of the neurons will be positive. And if it's very positive, then the output will be very, very positive. And this output will be used to connect to other neurons. So it's just a very simple computation of incoming data from other artificial neurons, so from other simulations. And what is important here is the weight of the connections between this simulation and other simulations.
So artificial neurons are basically small software mathematical calculations that are connected to each other.
Exactly, yes, exactly.
And then based on probability, it the where it ends up makes another probability and then moves the information further.
Well, you talk about probability, okay. Most of the models are not probabilistic in artificial neurons, but you're right, absolutely right, okay. Some people do probabilistic neurons, and we do sometimes also. It's fun, but but as far as I know, for instance, ChatGPT is not using this kind of neurons. But
okay, that's good. So artificial intelligence basically many mathematical formulas which talk to each other, and hopefully the outcome is interesting.
yes, and this comes back to your initial question about connectionism, about connecting different entities to process information. This is exactly what you defined.
And then I understand that because then I understand the 3D aspect from neurons is that I mean, it's sort of I don't know how much quantum physics is connected to the work your guys are doing, because what I've learned from quantum physics is that a particle can be there or not there depending on whether you look for it or not, which means that nature can simultaneously check multiple routes to come to an end conclusion.
Yeah. Well, in our case, in order to have this kind of behavior that you to exhibit this kind of behavior, you need to have coherence of the wave function.
Yes.
And this is hard to get. You have to have a system which is isolated, okay, no perturbation at all, okay. And one of the required condition, not sufficient but necessary condition, is to be extremely low temperature, for instance, okay, which starts very badly because we are standing at 37 degree always, okay,
Yeah,
to have our neurons, okay.
which is the temperature of life.
Yeah, absolutely. And you have to look at very small things, which is also very bad in our case because a neuron is about 20 micron of diameter. It may sound small, but it's not small enough by something like 6 order of magnitude. So, it's really wrong. So this said, okay, there are a number of, I don't know, theories which have been written about possible quantum effects in neurons, okay, and I don't know about the validity of these things. They are not mainstream, certainly not mainstream, those theories, but they've been around for quite a long time, since easily 40 years, okay. And as a matter of fact, we do not consider any quantum effects in our work. And this is the beauty of using biological neurons, okay? You don't need to know everything to start using them, okay?
No,
so
that's
if
true.
they have quantum capabilities, perfect. They come with them.
Yeah, that is the amazing thing about nature. Yeah. Now, I mean, partly I'm also operating in the agriculture sector where a group of farmers already are understanding that quantum physics is part of agriculture. Things are happening on such a small level that rationality cannot explain it, but you see the differences happening, and you do not. The only thing is for you to not to understand, but how to move with it instead of how to understand it. And then suddenly biology becomes so much powerful than what you would think from a mechanistic point of view. And yeah, cool that it happens. And but then how do you turn it into something that becomes as a computer power, like let's say biocomputing? How does it become computing with all the spikes and the electrical current?
So there are different ways to answer this question, okay? First, you will notice that you are yourself right now, you are able to understand what I'm saying. And so you are performing a computation, okay, in a very broad acceptance of the word computation, okay. So since your nervous tissue is able to do this, there is no reason why in vitro nervous tissue would not be able to do it, okay. So let me give you another angle to this statement, which are, again, artificial neurons, okay. They are able to process information, to recognize pictures. You can even discuss with them with ChatGPT. So the real one and the simulations are both able to process information and therefore to compute something useful. So this is your answer.
But how do you talk to neurons then? I mean, let's say that you have a interface where you can ask your biological neurons some questions.
You know, the thing is that I would say, if I may, it's not the right question.
Yeah, for sure. I mean, I'm not the expert. I'm just
The question is
extremely
not
curious.
how you talk to people, so, but how he can understand it.
Yes, for sure. Yes.
So, because I can tell you how you talk to neurons, okay, and it's very simple, and it's the same way your here is working right now, actually. So, if you send some stimulation currents at 1 hertz, 1 times per second, for instance, okay, to one of the electrodes, to which you connected a brain organoid, you are already talking to it. You are sending some information, okay, 1 times per second, okay. And this is what I'm doing right now with your audio system, okay. You're able to hear what I'm saying because I'm just exciting some of your neurons, Okay, so this is your answer. How am I talking to neurons? Very simple. Just send some spikes.
You then I mean, for me, I mean, it's information processing, and then I mean, I understand from a artificial neurons that it is mathematical, and it understands. It's sort of like voice recognition, and then based on what it has learned, it can come up with a reaction based on the database it has. To put it very basic. But if it's not mathematical, how do neurons then do the math of understanding speech and return that into speech?
It's correct. But this is a different question. Then it's not now how you talk to neurons, but how do you get a meaningful
Yeah,
answer?
mental track. Yeah, but that's the same. That's the same with current artificial intelligence. If you need to prompt it, I think that's the right word. The right prompts, and then it comes with with an answer that is useful. And otherwise, it throws out blurish.
No, no,
Oh,
no, no,
okay.
no. I wouldn't get this analogy. I wouldn't take this analogy here. Okay, because here there is the key word is training.
Mm-hmm.
So I can send some simulation to your hearing system. It will work only if you know English, for instance. Okay. So your nervous system has been already trained to understand English. And it's the same for artificial neurons. Okay, it will recognize a dog from a cat if it has been trained to recognize a dog from a cat.
Mm-hmm.
The same problem and the same solution. The solution is very simple in its principle, okay, is to change the connections between neurons.
Mm-hmm.
Okay, that's it. Just change the optical. You just find the right connections between the neurons and you're done. You know English and you know how to recognize a dog and a cat.
It's like it's amazing that it can be done. I'm so curious how the hell that is possible with a natural system, because there's also sort of this disconnectiveness from, I think from a humankind perspective. Like, how can biology do that? I mean, it's something you cannot control. You can steer it with hairs and frequencies, so that's how I understand. But how does it come back with an answer and make that answer understandable?
So if the question is how can a biological system update its connection correctly
Mm-hmm.
to provide a meaningful answer or to learn something? Okay, I am just sharing the same curiosity as you.
Okay, because
I have no clue. Okay, so, so
that is very clear.
so there is the literature gives us some directions. Okay, but just to be clear, you as a human being, Alexander, you are able to learn.
Mm-hmm.
Okay, so you have this built-in mechanism.
Mm-hmm.
Artificial neurons and AI is able to learn. It's not built in. We had to add this mechanism. So we understand it perfectly. That's the good thing. For us, unfortunately, it's like this, and we have to understand a posteriori how it works. So I told you, we know that it's basically the change of the connections between the neurons, okay?
Mm-hmm.
But it doesn't really answer the question. How do you change the connections between your neurons? Okay, it just and
Okay, but that part I understand from a chemical perspective. I mean, if
yeah, well, yeah, yes, but actually you're right. We can understand how to change a connection. It's just the number of synapses and the intensity of the synapse is going to determine the weight of the connection.
and a
Okay.
potential difference between the two connections, whether they attract each other or you can steer each other away. I mean,
Yeah. Okay. So yeah. So you may have it's connections may stop. Okay, synapses may stop, and indeed disconnect. Right. Now, it still doesn't answer to the very more profound question: is how our biological system knows which connection to increase or to decrease in order to learn something.
Yeah,
And
yeah.
we know it perfectly for artificial neurons because
Yeah.
people made it and invented it. And we have very few clues on this. And in science, experimental science, if you talk about biology, there are two very broad classes. There is in vitro and in vivo.
Oh, help me out. What is the difference again?
So in vivo, you are playing with monkeys and rats and mouses and
Okay.
human
Yeah.
beings. Okay. In vitro, you are going with pipettes and glassware. Okay. So training and learning has been studied for the past 50 years. It's called behaviorism. Okay. And it's been studied in vivo. And in vivo only. Doing learning in vitro with neurons has not been studied at all. So we are almost alone, a few of us scientists in the world, trying to do this, which has never been done before.
Yeah. Then I start to understand it. That then you give a certain spike, and then it answers with its own spike, and then you can play with that.
Yeah, well, you can interact, okay?
Yeah.
But that's not enough. You don't want only to interact with something stupid. You want it to learn something useful.
Is it then something like if it gives the right answer, that it recognizes a duck as a duck, you give it food or something as a reward?
So yes, indeed. So I told you at the beginning that two ways basically to stimulate the neurons. I told about electricity, okay, but there is another way that I've not discussed so far, and that you are citing here, which is to use chemical stimulation.
Mm-hmm.
And there are many chemicals to which neurons are sensitive. Okay, there is the broad range of so-called neuromodulators or neurotransmitters. Okay, which are going to directly or indirectly affect the synapses, which is
Mm-hmm.
exactly the connections. So the weight of the connections, exactly what we are trained to modify to learn something. So indeed, there is one mechanism which is called reward, which is very well known in behaviorism, okay, where people have shown that they can influence the learning of something by rewarding or punishing, okay, a living system. So this is also one of the protocols that we have to release, for instance, dopamine if our system, our brain organoid is doing what we expect.
Interesting. Um, there is another question that I think it's now suited to ask. Where then does the information get stored? Because it needs to understand that the next time it sees a duck, it's a duck. Does it store its learning somewhere? How does a neuron remember?
So it's funny in your biology. There are many things that we know very well, and many things where we have absolutely no clue at all.
Mm-hmm.
And but these are one of the things where there is some understanding. Okay, so I'm happy to say this. So in the connections, just this. Okay, each neuron is typically connected to 10,000 other neurons.
Okay, and
Yeah,
and that's in 3D.
yeah, yeah. It's complicated, right?
No,
Okay.
no, no. That means that there's a lot of remembrance by these different connections, because then you can store a lot of information by these connections.
Obviously, particularly if you consider that you have 100 billion of neurons.
Yeah, yeah, I know. But then it becomes much clearer that the connections, the type of connections, is actually the memory.
Yeah, it is.
and then it can turn a, I don't know, a 2D, or then it can create a 3D image in its own system of its own 3D system. And then what you said, then the different neurons switch on and switch off to create that image. And because you have so many, you have exponential large amount, infinity numbers of, how do you call that, connections that are associated with a certain image, sound, or whatever.
Yeah, it's not exponential, but it's big.
Yeah, I mean, but then I understand how, then for me it becomes much clearer how neurons learn and store, because if it's sort of like a sequence of synapses that are connected together in that
Yes.
case.
Yeah, that's right. Yeah.
And then if what you mentioned before is that not all of them spike based on the information they're processing, that means that you have so many combinations, and every combination is actually the corresponding information that's stored.
Yes.
So basically, it then becomes 0 and 1 in a way, if the spike is a point of recognition. But if
Yeah,
it's,
you're right. Yeah, the spike can be considered as a 0 and 1
I
quite
mean,
well. Yeah.
in that regard, I mean, what then makes me more curious is then how the chemical information flow is being used to. Is that also be then, to rephrase that into a question? Is that also a way to store information, or does it has a different function?
The flow of which chemical?
Yeah, the information flow is either electric or chemical, what you mentioned. So what is the purpose of the chemical?
You mean the neuromodulators?
Yeah.
So there are different types. Um, so there are some chemicals like neurotransmitters. So I'm going to talk about the glutamate, for instance. You can eat glutamate, by the way. If you cook Chinese, you are going to eat glutamate. So it's a neurotransmitter. Okay. You will eat it as a salt. Okay.
Mm-hmm.
The glutamate is going to bind with some receptors in the synapse and will lead the neuron to spike.
Okay.
So if you ask me what is the effect of this particular chemical substance, is going to make neurons spike. Very simple and very well understood. Okay, to the molecular level.
Mm-hmm.
It's a dream situation. We understand something. Okay, so in neurology, it's fantastic. No, but as soon as you get into the details, we understand more and more. The problem is when you take some distance, it becomes more complex. So now I've been talking about one of them, okay, and one of these components, which maybe is the less interesting one. So let me tell you about something we don't understand,
Mm-hmm.
which is a neuromodulators, which is a bit like a neurotransmitters. It's still a big molecule, okay. Well, it's not a big molecule; it's a small molecule actually, which is called the dopamine.
Mm-hmm.
We know that the dopamine is implied in the reward process.
Mm-hmm.
And we know it has indirectly some effect on the synapses. Because when you have a pleasurable experience, you are likely to make the situation that lead to this experience reproduce again. Okay, so you're going to learn a way to reproduce this, and it's a mechanism of learning actually. How these neuromodulators is going to magically, I would say, update the weights of the connections between the neurons. This is not known. We know that there is a link. It's very clear. But we don't understand the link between the learning and the dopamine.
No, I then assume that there are various transitions steps
Yeah, you're right. Absolutely. There are various.
From a chemical point of view that extends into different areas where you do not understand what the cause and effect is.
Yeah, yeah. In biology, you often talk about cascade of events.
Yeah.
Okay, so it's very classical. Okay, we used to have cascade everywhere. Okay,
Mm-hmm.
but this one is really not understood at all, and unfortunately, this is absolutely fundamental to what we try to do.
And the better you understand the cascading within and between neurons, the better you understand where they store the information, the better you understand how you can interact with neurons.
Yes, yes. In order to make them learn something.
When becomes when what is the tipping point when learning becomes a reinforced loop that it starts to learn by itself?
Um, I don't know.
Okay. No, yeah, because I mean the way we describe it, and basically the way you are describing it, how it functions in our brain I assume it's the same way. Then if you have enough connections, you can understand cause and effects suddenly. I mean, I see it with my child. In the beginning, it's quite linear, and when they arrive at a certain age, they start to understand cause and effect, and then start to interpret the situation based on the knowledge they then have, and then you see them evolving based on the information amount they have of the experience they have. But then that's then also curious because I understood there are different types of neurons, and you explained that in the beginning. Connecting these different neurons together, does that also mean that the information storage is differently? Because what I understood, we have a synaptic nervous system and a parasynaptic nervous system, and then there are different things happening also with dopamine from a hormones perspective. Is that also happening in the neurons you're using? And is one neuron happier with to have certain neurons around him, or do they tend to move together and say, okay, you do not belong here, we do not want to connect with you?
So, what I can say from our own experience and from the literature, it's incredible how neurons like to connect each other, even if
Mm-hmm.
they are absolutely not from the same area of the brain. So,
Okay.
you take, for instance, the hippocampus, which is one specific area of the brain implied in a memory, and you create a brain organoid of the hippocampus. You take the cortex, for instance, and you create a brain organoid from the cortex. So you have two small balls. Okay, you put them in physical contact, and in 1 week they will create connections spontaneously. So I've never seen such things that um, neurons that wouldn't like to connect, and I never read some things like this. They love to connect. Actually, I even read some papers about interspecies communication from human brain organoids to rat brain.
Mm-hmm. But then does it then matter that you use human stem cells, or can you use any animal stem cells and have also a neural system working?
Yeah, there is no striking difference between the neurons of rats and human.
And why are you then using the humans and not of another animal?
Because when you create a company, you never know if you are not going to discover something unexpected. Okay.
That sounds like a real inventor. Yes, serendipity is something you need.
Yeah, well, if I discover something unexpected, which has medical applications, for instance,
Hmm.
it's better that it's human than for rats.
Okay, clear. Because that makes me curious. Then I mean, it gives two questions. One of them is, what would happen if the different neurons from the different parts of our brain connect to each other in a different structure than our brain. We know that the cortex is the front part of our brain where we humans have evolved our rationality, if I think it's called correctly. And then we have somewhere the hippocampus where our prehistoric experiences are stored. What will happen if you start to mix these types of neurons together? Does it generate a different learning path for itself?
This is exactly what we hope.
The match and mix is what you guys are discovering at the moment.
Yes, exactly. And we can get some inspiration about morphology of our brain and the brain of mammals in general. But it's a lot about trial and error.
That is then, because the reason I'm asking that question is that every part of the brain, in my perspective, has a function and has evolved in that part. For instance, with a sympathetic part of the neurons that's triggered on fight or flight, that seems to be their the janine, their natural state, and the other ones are more trained to work for the nervous system of our indigestion or our let's say our muscles and that kind of things. I mean, does that also mean that these neurons have a sort of presetting based on evolution?
Yeah, we believe so. Well, not that we believe so, is that the literature seems to show that for the different areas of the brain for which have been identified specific functions, the neurons' characteristics are indeed different. They have been measured, by the way, by the professor Markram from in Switzerland. So you can go actually online and you have a sort of atlas of all the types of neurons of the human brains and what they do and how they work. So, and yes, we believe they are all different and for each area of the brain.
Then the challenge is finding the right quantity matching with the right species to turn it into something that is useful as a computation you call it interaction system. How long? How much? What is the time frame to have something meaningful?
We hope to have mastered the training side of brain organoids within the next 5 years, and then to have a server online able to serve customers in following 10 years.
Because is it now still in baby or an infant, or still an embryo?
What do you mean? Well, no, yeah, I know. You have to imagine how is the lab. So the lab just below here, okay? And maybe I describe what we have, okay? So right now we have in the lab about several thousands of brain organoids, okay, at different stage of maturity, okay? And we keep them about 2 years in the lab. And we are going to put them on electrodes each time we did them. So I was just answering to a question about the evolution of our neurons, you know. So you have to understand that in our lab, which is sitting just below here, we have several thousands of brain organoids.
Yeah.
Okay. And some are like 2 years old. Okay. So they have different maturity.
Oh yeah.
And we take them, put them on electrodes, and make experiments with them.
That's how they learn, because then they, and that's all a community, or is that it's very separate?
No, I want to say it's a community because it's more like we take them, we put them on electrodes. They are going to live for between a few days and few months, and then they're going to die. Okay, and then we need on some others.
Mm-hmm.
So it's more about making experiment to find the right way to modify the connections in order to learn something.
And what happens if you can keep them alive for a longer period? Because then you can train them longer.
Yeah, of course, you're right. The longer we can keep them alive, the better it is.
Yeah. And is that the end goal? Is that the necessity of the end goal, that you find a way to keep them alive in order to have the information stored in the 3D model?
We hope that it's not necessary to keep them for a month alive in order to learn something. Okay. At the stage where we are right now, we just want to demonstrate our ability to reprogram some brain organoids. But you're right, to make something useful, okay, it's not only, you cannot limit yourself to something that lives a few months, okay? We have to live for a few years. So after we've been successful in training, we'll go in extending the lifespan of our brain organoids. Our neurons as human can live 80, 90 years, no problem. We live with the same neurons, okay? So we know it's possible.
How do you make it? Because I don't know if I'm over asking you, Fred, for the next one. Because recent research showed that there is a microbiology community living in our brains in synergy. Is that also something that in the future you're looking into? Because microbiology tends to interact with everything in our human body, and the research is suggesting that might also be connected to the way we learn and the microbiology in our brain. There is a connection, but we do not really understand what and how. But there is a reason why they're there, and there's a reason why our brain is collaborating with them.
Which microbiological organism are you referring to?
I think they say that there are types of bacteria living in our brain, and from a beneficial point of view, not from a infection point of view. I mean, it is, and next to that, they also have found that there is microplastics in our brain, so that's a different thing.
So you refer to the microbiome?
Yeah, our brain microbiome.
Yeah, I'm not aware of this. I don't know anything about microbiome.
I'm just asking because it's interesting where we're currently finding the microbiome almost everywhere in our body, and I think this was the last 2 years or the last year that they figured it out.
It seems to be still at the stage of hypothesis and discussion, right?
Okay. I can assume that they have not opened up a brain currently and did DNA analysis, or they did on deceased people. I don't know.
Yeah, actually, Wikipedia says that it's a hypothesized microbiome of bacteria. So sorry, I didn't know about this. But the thing is, the good thing about using living neurons is that if there is a microbiome, maybe we'll get it for free.
Yeah. Yeah, and maybe it does the electro what do you call it, the hormone reward system automatically without having to put that into the neuron system. So you interact with the bacteria to produce the hormones you need to reward to reward the neurons. I have no idea.
Well, so typically if you talk about dopamine, for instance, for rewarding, the way we use it is that we just buy it. Okay, you can buy dopamine.
Yeah, but you have to add it into this. You have to put it into the fluid, and then
because you're right in the sense that in our brain there are some neurons who I would say create dopamine.
Really?
Okay. Yeah, these are dopaminergic neurons. These are the names of those neurons. Their goal in their life is to create dopamine.
And I assume that they also create the other hormones.
No, dopaminergic will create only dopamine.
I mean, I think there are. are there also other hormones in our brain based on our experience?
So I'm not sure these are hormones, but
Okay.
yes, you're right. There are many categories of neurons. You have glutamatergic neurons, for instance, that will create glutamate. Okay,
Amazing.
so each type of neuron is going to create a different kind of molecules, which will help to discuss or to interact with other of their friends' neurons.
And that means that once you understand in the first phase where you're now in that you can interact with the neurons, then you can add these kind of neurons there, because then you don't have to add the dopamine into the system, but the system can
So, that's no, but that's a very good remark, because there has been a publication last year, very interesting, in Vienna about this, where people were actually doing an assembloid, okay, using dopamine neurons, dopaminergic neurons, to create the dopamine, okay. But now, but these people are neurobiologists, okay. I'm not, okay. I'm just an engineer, okay, let's say. So as an engineer, you ask me, well, you want to use something as sophisticated as neurons only to create dopamine that I can buy for $100, I'm going to buy it, okay? And I'm going to master the release of dopamine. I'm not going to rely on biology to do this job, because this I can do this. I don't need biology to this.
Yeah, and that means you have a little bit more control of what happens in the system, in the box. And
Exactly. Yeah.
if you have a dopamine releasing neuron, you need to control that one also with the system you're already controlling. But it would be interesting as a hypothesis what will happen when you have these also in the system, but then we move basically maybe to self-consciousness and self-awareness, because then it can reward itself or punish itself
So,
depending on its
but
experience.
I encourage the listener to Google assembloid
Mm-hmm.
dopamine Vienna, okay, the city of Vienna in Austria, and they will find this publication of Academy of Science of Vienna, who has done a wonderful work published last year about assembloids, in particular using dopaminergic organoids. So
Amazing.
one of a kind work. So
And there
very
is
interesting.
also one thing on my paper which, based on what you're describing, I have to ask. I mean, what is your how does your technology compare to Neuralink? Because there they want to hook up a human brain, which is like the real situation of neurons in its natural environment. Let's put it that way, and then connect it to a, I don't know, maybe a 0 and 1 interface. Um, how does that differ for what you guys are doing?
Well, first, the common point,
Yeah.
okay? The common point is that Neuralink and us are both interacting with human living neurons. This is exactly when we join. Where we make the difference is that we are in vitro and they are in vivo.
Okay.
There's a very, very big difference, because, and particularly because they are the top of in vivo, which is human being. Okay. Um, there is a question of also of scale. Okay. We have not been talking too much about this, but the size of the brain organoids that we are using are small like this. I mean, you can see them, but they're very small. Okay. Neuralink is working with a real brain, okay? So their electrodes are way, way larger. In our case, they will be unusable.
Mm-hmm.
They are way more too big, okay, for us. The way of collaborations exist indeed. For instance, for individual electrodes, so the point of contact, we can test its biocompatibility on a specific human brain. We can test it. We can do even better. If you have a patient, we can take some of his skin, create stem cells, create neurons, create brain organoids, and test if the electrodes is compatible with its own neurons.
That one went too quick.
Specialized medicine, okay? When you try, in our case, okay, I told you about stem cells at the beginning. but I didn't tell you how people create stem cells.
That's true.
So in order to create stem cells, we take skin cells.
Really, I thought you would take them at the bone marrow.
No. That's what's new, actually. Therefore, what is interesting, so from skin cells, there is a way invented by Professor Yamanaka, Japanese Nobel Prize, to create stem cells.
Wow.
So what is interesting is that, of course, the stem cells has the same DNA as your skin cells.
Yes, true.
Therefore, all the neurons and everything that you're going to do are going to have the same DNA.
Yes, and then every DNA is unique.
So if you, Alexander, I wanted to test the compatibility of my electrodes with your own neurons.
Yeah.
What I could do is take some cells of your skin, create stem cells, create the neurons, create the brain organoids, and test my electrodes on your own neurons.
Whether they are compatible or not.
Correct.
And then it's also then a connection with specific DNA. Some DNA will probably function better than other ones.
No, but then we have to adapt. Okay, of course,
Yeah.
you may be right. But then I may have to adapt my electrodes to be compatible with your neurons. Science has to adapt to human beings. It's the way it works.
Do you then apply that the natural frequency? I mean, that's moving a little bit into quantum physics. That the natural frequency per DNA, whatever entity, is different.
So, well, if you talk about the kind of interactions that we have with brain organoids, okay, they are done by programming. Okay, we program interactions with programming language like Python. Okay, if we take two brain organoids, even from the same DNA, so these are perfect twins, if you want. Well, they are not so perfect. Okay, I can tell you, each brain organoids that we have are different.
Yeah, and that's nature.
And so if I had another DNA, they would be also different. So anyway, we have to develop ways to interact with brain organoids which spontaneously adapt. At the beginning, we recognize the characteristics of the brain organoids and we adapt the kind of interactions we are going to have with it.
You mentioned that we were not talking more specific about the upscaling challenges you have, because you're an engineer and you're and also as a co-founder, your goal is of course to turn it into a scalable product. So what are the challenges now to get to there? I mean, what TRL level are you on, and what is needed to get there? I mean, the reason why I'm asking that, I know from a lab-grown meat, I've talked to some of them, and there they were able to turn stem cells into meat kind of cells or fat like of cells. How are you playing the game at the moment?
So playing the game, there are many ways to describe this. Okay, so are you asking from a science perspective, from a money perspective, or from a communication perspective?
If eventually all these three are linked together, because if the one doesn't function, the other ones will also lag behind.
Yes, you're right. They are interlinked. Okay, from a science perspective, we are advancing on a number of different strategies to train the neurons to learn. Okay, this is really the main thing. We also focus at the same time on lifespan expansion. On communication, I'm talking with people like you to have the maximum number of people hear about what we are doing, because biocomputing is so new that nobody knows about it, including investors, which brings me to the last part, which is the financial side, to have more awareness about this new industry which is coming.
But I can assume an investor who is tech savvy and who understands the next frontier, they will be eager to join you guys. Because
That depends. Okay, that depends on the kind of investors. Okay, if you take a venture capitalist, for instance, they have they are going to consider how well this field is known or new.
Mm-hmm.
Okay, so you have a number of VCs, venture capitalists, investing into quantum computing, for instance, today. Okay, but quantum computing has been around for 15 years. Okay, and biocomputing for 5 years. So it's still a bit early stage
Mm-hmm.
to get VC funding. So it's more about getting some very wealthy individuals from sovereign funds of some countries. I think at short term it's this.
And when is your next funding round then?
So it when we find the right investors. Okay. Uh, we
We're
are
laughing about that, but for me, that is so evident because I know a lot of stories where they had the wrong investors and that those companies do not exist anymore. But finding the right investor is not easy.
indeed. It takes some time. Um, Hopefully, my colleague and I co-founded another company that is called ALP Vision,
Mm-hmm.
ALP Vision, which works in a totally different field, but connected to our PhD, and is quite profitable since many years. So we have the money to invest into Finance Park with our.
Wow, that's luxury.
Yeah, it's a big luxury.
No, that means you have a revenue stream coming in which is predictable. Um, and then means that you can know when you need more money, either from your own company or you get the slow investors in. And what is the profile of an investor? Just as a shout out, who knows who's listening?
What is
The
the
profile you're looking for
for the future investors?
for those who want to join your ride.
Yeah. Well, those who discuss with us, we have all horizons. Okay. Few in Europe, mainly in US and in Asia. So a little bit in Middle East. Um, it's about what we're looking for is really patient investor who are really uh, interested by deep tech. I said really because there are too many claims about doing deep tech today, which is not deep tech.
Mm-hmm.
Okay, or let's create a class for us, which is called would be, but the same class as fusion and quantum computers. That would be hardcore deep tech.
No, yeah,
So,
no, definitely, and it takes a lot of exploration to figure out the right path forward, and that even can be the wrong path, which you only discover along the road, because you have so many choices to make every time, and you never know whether it's a good choice or not.
that's the problem in general with life.
Yeah, yeah, no, but also if you're so fundamentally figuring it out, it's sometimes two steps forward and maybe sometimes three steps back because the direction you're moving is not the right one. We've missed something out, and then you need to re-engineer where you took the wrong turn.
Yeah. This is the deep tech aspect. Okay, which is very demanding for
What is
investors.
deep tech? I'm nodding. Yes, yes, I understand it, but I have no idea what the definition is of deep tech.
Oh, well, I'm not sure I'm better than you on this, okay? But it's deep tech is really opposed as if you're doing, I don't know, Uber, for instance,
Yeah,
okay? It's not deep tech, okay? It's just software engineering,
yeah, yeah.
okay?
Agree.
Deep tech is really when you go deep in the technology, literally, okay? That you rely on fundamental research on things which have not been done before.
And they call that
Okay, not been done before on the scientific perspective.
deep tech. I would say that's fundamental tech.
Yeah. There is this word deep is really um, we used a lot, and you have deep learning, for instance. So it's the fashionable word.
Currently. Okay, then I understand what deep means. And is there any are you still looking for people? And what kind of people do you want?
So I would be happy to hire way more people. Okay, we are 6 people right now. I would have to have 60. Okay, um, and would be neurobiologists, engineers, and data scientists. Data scientist, that means AI expert. Okay, but I don't have enough money to hire all of them.
No, no, no. I understand it, but who knows that somebody is listening and then is curious whether this is something for them. And if you have vacancy, they just can call and see whether it fits or not.
Yeah, that would be wonderful.
Because upscaling is always a challenge. That's what I've learned with all the conversations I'm having.
Yeah, but it's always my hope, discussing with people like you, is to find some people who are really sincerely motivated by the project.
Yeah, because that's what you need in this phase. Otherwise, it's not working or will not work. Is there anything, Fred, that I missed out on our conversation? Because I've learned a lot, new things, and I didn't realize how interesting, spectacular, and I don't know how the future will develop if biology finds its own consciousness, but that's topic for up for another conversation. But is there something else that we missed out that you really want to share that is important to the whole journey you're walking through?
There is a number of exotic things, okay, I would say. First, there is all the thinking about ethics and philosophy.
Mm-hmm.
And so we have a number of discussions with philosophers, so we don't know anything about ethics, okay? But some people know, okay? So there is going to be a number of publication in the academic community about what we are doing. We bring the science, and they bring the thinking. So
Yeah.
so this is also one thing that we try also to build, okay? To build awareness in the society about biocomputing, what it is, so people are not afraid of it, okay? We have a lot of, so if you go on a Final Spark website, you will see that there is a section that says live. If you click on this button, you will see the activity of our neurons in live,
Yes.
24/7.
It's quite weird to see that.
Yeah, but we are the only one in the world doing this. Okay, so it's very funny. What is even more funny is that well, of course, there are many universities with whom we collaborate. Okay, and they have access to our neurons, and they do experiments 24/7. Okay, there are even companies renting access to our neurons now to do some research.
That means you have a web interface with your neurons.
Yeah, we have a full API that enables to do the web interface with the neurons, indeed. And we have a GitHub repository. We have a Discord server. So if people after this talk want to connect to Discord, they can come, just ask questions. We'll answer. We answer to all questions, and it starts from students, age 10.
Cool.
So we say some. Well, we try to answer to everything. Okay, so that's really fun. And people are nice. It's not very controlled server where we don't ban a lot of people. It's quite okay. And there is also something else which was unexpected, but I think it's good for people to know that there are some artists that connect to us. Okay, and they say they want to do art by connecting with our living neurons. So you can find on the internet a number. If you just tap art and Final Spark, you will find some things. There was a painting, an interactive painting that was done by a Zurich team in Switzerland a few weeks ago, which is still live for 3 days in Switzerland. Okay, it's incredible thing. It's like a painting, which is basically like, yeah, you are in front of like a synthetic skin,
Mm-hmm.
and you can push with your hand on this elastomer thing, and it's going. By doing this, is going to send some simulations to the neurons in our lab remotely. and the painting is going to deform as our neurons are answering. So you can interact actually with your hand and feel the response of the neurons remotely. I mean, from a scientific perspective, there is absolutely zero here, okay? But from an artistic performance perspective, I thought this was interesting.
No, I think art and science is so well. There is a very thin line between art and science. Because artists can be scientists without knowing that they are scientists, because their curiosity pushes you into certain areas where you never thought an interaction would exist. I mean, I've been in the past with biohackers community, and what they have done with things is still resonates, and I still remember that. And it's they're very. I mean, there are physicists between them. They turned into artists and playing around with existing technology and turn that into something spectacular, and also challenging the way we see and look at things. That's always useful when you have artists in your network, that they challenge the way you look at technology, because they see it in a different application. And lastly, then, is there anything else you want to add, Fred?
No, there would be many things to say, but I'm going to stay
Because
at this
I
point.
just remembered a science fiction series from Australia which was called Farscape. I don't know if you ever heard of them.
Farscape? No, I don't know.
They used a living spaceship as a spaceship, and it could restore itself depending on whether it had any what do you call that, when it was broken or something. And they had actually sort of symbiotic machine biologic robots walking through the spaceship. And the way you're explaining how the interaction with neurons is, I'm also taking that to what if you could also use biology as a skin or something which you could integrate into your in our technological world. Then biology becomes much more closer than where we have put it today, because we have turned so mechanistic in a lot of things.
That's very funny that you say this, okay? Because I think exactly the same thing, okay? And describe exactly the same thing as you said with the same words as you use,
Really?
okay? So there's a nice coincidence here of thinking. I was thinking, because this is pure science fiction, okay? Imagine, so you are sitting here in a room, okay, and around you you have basically either artificial objects or living objects.
Mm-hmm.
Okay, you may have some plants or flowers, okay, or cats, okay, and the rest are artificial. So there are two classes of objects: artificial ones and living ones. Period. Okay, there is no hybrid. Okay, no artificial hybrid. Okay, but I can imagine a world where there would be artificial hybrids. Okay, like you take a glass of water. Let me describe how it could be a hybrid glass of water. Okay, it looks like a glass of water. It's still transparent. You can still put some water into it. Okay, but this has some receptors and neurons which are able to detect what the gas that you expire
Mm-hmm.
and your breath, and analyze it and synthesize spontaneously some molecules which are good for your health. And when you touch it, it adds also some information to the correct formulation of molecules is going to release in the water, which is good for you. So it's kind of bidirectional communication. So it's not totally a glass. It's not totally living, just in between. Okay. This kind of things, this kind of hybrid species, I think it's an interesting direction. Pure science fiction.
Okay, thank you, Fred, for the conversation.
It was a pleasure. It's always a pleasure.
I'm pleased you took time to listen to the whole episode. Why not consider joining my Patreon to support me in creating more of these conversations? Or even rate my podcast in your favorite podcast app. Thank you again for listening, and join me next time.